start the stream. Okay, there we go. Okay, so I'm very happy to introduce Orit Peleg. And for our second to last biotheory seminar for the year. So Orit is a very interdisciplinary researcher who's sort of the interdisciplinary nature of her work is borne out in an extreme way in terms of her sort of previous affiliations. She did a bachelor's and master's in physics and computer science in Israel. And she went on from there to do a PhD in material science from ETH Zurich. And then she went on to do a postdoc at Harvard, where she worked with many folks, including Maha. And then she started as a professor in the Department of Computer Sciences and also affiliated with the Biofrontiers Institute at the University of Colorado Boulder in 2018. And Orit has been interested in sort of the collective computations of groups of organisms. For instance, one problem, as I understand it, is trying to understand how swarms of bees composed of many members retain their structural stability in the face of mechanical stresses, for instance, imparted by the swaying of a tree branch. So with that, I'm very excited to hear about physical computations in insect swarms.
Great, thank you so much for the lovely introduction. Let me just share my screen and hopefully you can see that. Yeah, so thank you for the introduction. Thanks for inviting me. I'm excited to be here today. And I'm hoping to tell you about some of the work that we do in my lab about physical computation in insect swarms. Even with a nice introduction from Karna, I think I might spend just a few minutes giving you a slightly more deep detail about the kind of questions that we work on in my lab. As Karna mentioned, I'm interested in communication in complex systems in biology. I think that as humans, we kind of get used to being able to communicate with each other over long distances without any issues. We have lots of different ways that you can do this, including the Zoom meetings that we are having right now. But as you all can probably appreciate, animals have to solve similar challenges using only natural resources that they have in their nearby environment. So in the context of communication, animals tend to use their bodies or some extended phenotypes. They use tactile communication, visual, chemical, vocal, and actually different combination of all of these modalities. And the regime of communication that I'm really interested in is not just pairwise interactions, but also communication in very large groups. So just to name, there's lots of beautiful examples. I'll just name two to set the stage. So one example of this collective communication is on like microscopic scale, slime mold that I'm sure many of you are familiar with. All these white dots are food sources. The slime mold is recruiting with chemical communication. And essentially, they actually also solve the shortest path problem that mimics the transportation network around Tokyo. So in a sense, they're kind of solving a physical problem of finding the shortest path that connects all these different food sources. Another example on a different length scale are flocks of starlings. I'm sure we all are also seen these beautiful movies of lots and lots of birds that are able to, using potentially pairwise interactions between each other to coordinate their movement and make very sharp turns and move efficiently in space. I kind of come to this range of biological problems with training in physics and complex systems, where actually this kind of emergence of collective behavior from pairwise interactions is very common. We think about it a lot, right? Atoms that are crystallizing into lattices and also larger non-living assemblies and emergent collective behavior. And I'm kind of using that as an approach to also try to understand these living systems. So in particular, I find myself thinking a lot about detectability, how signals are, can have more or less signal to noise ratio, compression of these communication signals in terms of minimization of the bits to represent information, robustness, how robust the communication network is to failure of individual nodes. And the approach that we use in my lab is we develop mathematical and computational models to get an intuition to the system, and then make some testable prediction that then we test either via collaborations or mostly actually in-house with behavioral experiments. And then we kind of iterate between the experiments and the modeling until they somehow converge. They don't always do, but that's the kind of the hope in each one of these projects. And the model system that we usually use are insect swarms, usually fireflies and honeybees. So that's what I will focus on for this talk. I don't know if you can appreciate how cute they are when you look up close to them. I think they are, but they're also really just remarkable in terms of their ability to communicate in very large congested groups, and that's kind of why we work with these model insects. So, okay, what I want to do today, if I have the time, I'll talk about two ranges of communication in large insect groups. The first will be in more congested environments, like in these honeybee swarms, where the individuals they're not flying, they're actually holding each other, they're holding hands to create these large swarm structures. And then I'll switch to also talk about more sparse swarms, in particular swarms where visual communication is being used with firefly swarms. Okay, so I will start by diving into the behavior of bees and some other dense aggregations. And I just want to echo what Karna said before, that if you have any questions, if you have any comments, feel free to stop me. I'm very happy that this would be more of a discussion than just me speaking, but as you like, feel free if you have any comments or questions. Okay, so bees. This is a story that started a while ago when I did my postdoc with Maha, and actually came from a background in material science, modeling biological materials on a very different scale, very microscopic scale. I never really did experiments before, and that was one of the beautiful things about working with Maha, that he suggested working on these biological materials with experiments, but on a much, much larger scale. The experiments are simple enough that I could actually do them. So these are the biological materials we worked on. These are this is a movie of a honeybee swarm on a windy day. As I mentioned, the bees are not flying, they're actually holding each other, and there's nothing but bees and a tree branch here. And as you can see, this is, it can get mechanically perturbed by environmental factors like wind. If the swarm breaks, that can actually be catastrophic for the bees. They might die from hypothermia, because one of the reasons they actually form these structures is for thermoregulation purposes. So we wanted to understand how can it be that this structure is stable, given the fact that there's so many individuals, there's tens of thousands of individual bees here, and a bee can potentially coordinate its activity with neighboring bees, but certainly not with bees at the completely other side of the swarm. So to do that, we started this. It was really an exploratory project. The idea was simple. We just wanted to create a swarm in semi-lab conditions, shake it and see what happens. Kind of, that was the the gist. So here is how a typical experiment look like. We have the swarm attached to a wooden board. You can kind of see that it's a jiggly here. It's an elastic structure in a sense. And we continue shaking them for a little while. It was very exploratory, as I already mentioned, but actually it turns out that from the response of the bees to that perturbation signal, we can learn something about how they maintain mechanical stability. So what we found out is that when we shake the bees for a while, bees start moving around in the inside the swarm, and they collectively change the shape of the swarm. So what I have here is a sped up version of that experiment. You can see the front and bottom view. The signal on is going to appear here at the bottom. So basically, they start with a cone-like structure. When we shake them, they spread out and they become more flat in a sense. And then when we stop shaking them, they go back to their steady state configuration. So then relating this back to modeling of these materials, we were able to model the system as an elastic materials of spherical bees connected by elastic springs, and relate the behavior of the flattening of the swarm to local mechanical signals. There's probably many mechanical signals that could explain this. What we focused on was the stretching of bonds that each individual bee is experiencing with its nearest neighbors. And since we know the bees are kind of moving up towards the attachment surface, we were able to program these bees in the simulation to also follow similar mechanical cues and basically go up the strain gradient in a way that also gets the swarm to flatten.
Can I ask a quick question?
Yes, Ben?
Do these results depend sensitively at all on the nature of the connectivity between the neighbors? So do you have like everyone connected to all their nearest neighbors, or?
Yeah, exactly. Yes, I mean, we tried a few different configurations. We tried cubic lattice, hexagonal lattice. That would change the number of connections you have per bee, and of course, this whole thing would heavily depend on the connectivity. So we made the assumption that it's isotropic, so there's the same number of neighbors in all directions. But actually, that brings me to I think my next slide, which is when we did these experiments, we had no idea what's going on inside the swarm, what is the connectivity. And now we actually, one of the heaviest piece of equipment I have in my lab is an X-ray machine. And we are using that to try to understand what's going on inside the swarm, potentially to also answer questions like what is the connectivity, but at the moment, we're just looking at things like density. So I'll show you some of these examples here. Basically, we put the swarm inside between the detector and the emitter, and by rotating the swarm, we can basically do a 3D reconstruction of the density inside the swarm. And one interesting thing that we found about at least the density and the arrangement of bees inside is that one of the assumptions we made in the previous model is that it's homogeneous, and it's not. So it's this is just a rendition of the three-dimensional reconstruction of the swarm. We can start looking more carefully at how bees are distributed, and in particular, we were kind of wondering, could it be that the bees following similar scaling laws that we know that individual organisms do. One example of that is strength to body mass that weightlifters experience. So this is an older paper from the 1950s, showing that there is a scaling law between the strength. So what is the weight, the maximal weight that an individual can lift as a function of its body weight. So we were trying to understand something similar and see if there's an interesting similar trend also with honeybee swarms. And it turns out that there is something. So we cannot really completely disentangle the weights, the weights and the individual here, because they're all bees basically. But what we can do is look at individual layers in the swarm, calculate their weight, how many bees are in each one of these layers, and then contrast that with how much weight they're supporting underneath. And we see a really interesting scaling law where the weight underneath each layer is scaled with the mass of that specific layer. And it actually doesn't depend what is the how many bees we have in the swarm. That's kind of, it seems to be a universal property that is true both for smaller swarms with just a few thousand bees and larger swarms with 10,000 bees. So this is a very new work we just posted that preprint on the bioRxiv. Yes, Avinish, I see you have a question.
I have a quick question. So with this scaling law, would you have gotten something similar by just, so what would be the naive interpretation if you just assumed let's say a hemispherical homogeneous surface?
Right, so we can actually relate that the shape of the swarm to that exponent. And if it wasn't that exponent, then we will get we can get a range of shapes, right? We can get just a cubical swarm. In a sense, it's kind of like a cylinder. We can get something that is actually narrower, close to the attachment surface, and becomes wider. And these are, in a sense, non-physical because they don't really minimize. Well, first of all, they're not stable mechanically, but also they don't minimize the requirement to minimize the surface area, which is coming from another functionality of these swarms that I just briefly mentioned, which is thermoregulation. So in a sense, yeah, we know that the bees are arranged in a half a circle or a cone-like structure, but they also do modulate the density so that the density of the bees closer to the attachment surface is higher in comparison to what's happening below. And I see that there is another question.
Yeah, so I was just wondering if you can pose this as an optimization problem where you select the shape so that the thing I would assume is that if all the bees are connected to the wooden board, is the most mechanically stable, but they will be vulnerable to thermal shocks. So you could write it as a thermal plus mechanical stability problem and solve for the shape. And
yes.
Okay.
Yeah, so perfect. That's a great question, and that's actually the direction we are trying to take this to. So at the moment, we don't really know what the balance is between the balancing the mechanical forces and the thermoregulation requirements. But we're doing some experiments in the X-ray where we actually vary the external temperature that would change the restriction on the surface area in a sense. And I think that would maybe be a way to connect it to something like you just suggested with an optimization problem of how the bees balance these two contributions.
Yeah, and another thing is that what I assumed these bees do is that they make a honeycomb structure made of wax and they hold on to it. How is this different from that kind of what you see in nature? Because this doesn't seem very biological. So when do they do these kind of swarming and holding on to?
Yeah, great question that I just didn't talk at all about this topic. So first of all, there's some bee species that actually build their nest outside, and when you look at it from the outside, it looks like a swarm, like I'm showing you now. But actually, underneath, in between, the bees, there's a honey comb wax structure. So these are not the same bees that we're working with here. These are European honeybees, and they usually nest in cavities. So during their productive cycle, they actually move away from one nest, and then a group of bees just hang, from a tree branch, and then they search for a more permanent position for their nest, and then they move to that. So, with the bees that I just mentioned, that I'm talking about here, there's no wax inside the swarm.
Oh, okay. So, sorry if I'm asking too many questions, but
no, it's fine.
I had another where, how do they cross-link with each other? Like, do they hold on to each other with their legs, or how does this work?
Yeah, so they hold on to each other with the legs. They have 6 legs. They have little hooks, and they also have hairy patches, kind of like Velcro, that they can also attach to surfaces. They can also bite onto other, individuals. So potentially each individual can have 7 connections initiated by that individual.
Oh, okay. Thank you. Thanks.
Is there another question? Okay. All right. So I think, right. Okay. So everything I told you so far was about the mechanical stability of a swarm. The bees are already aggregated, and they're already forming a coherent swarm around the queen. But one question that we were trying to answer recently in my lab is, how do the bees become a coherent, aggregated, dense swarm in the first place? So we got some clues from some demonstrations that beekeepers do, actually, and we reproduce it in the lab. So I'll play this to you, but before, let me just orient you. So this blue box here, that's our commercial beehive. The queen is already inside. There is a very narrow slit here that I'm highlighting with my cursor. I hope you can see. It's just, that's the entrance to the hive. And we basically dumped the rest of the bees a little bit further away from the entrance. And this is what happens. Somehow the bees know what to do. This is sped up, and you can kind of see them walking towards the entrance where the queen is. And if you look a little bit closer at what's happening at the entrance, we see that there's a lot of bees that are fanning their wings. If you could hear the sound, it would also be very obvious. If we zoom into this behavior, what's happening actually is that the bees are extending their abdomen. They're kind of pushing it upwards, and they're opening a pheromone gland called the nasonol gland. This is this white strip over here that potentially the bees are using to communicate with each other about where to go, giving some directional information. So they produce that signal, they sense that signal with their antenna. And in a sense, it's kind of a similar problem to a microscopic problem that I'm sure many of you are familiar with, of aggregation of social amoeba that also produce and sense the same chemical signals. But the bees use a trick that the amoeba, as far as I know, don't use, and that's airflow. So the bees, because they fan their wings, they actually direct the signal in a particular direction, actually away from their head, so kind of backwards. And what we noticed just anecdotally from some experiments is that a bee would potentially sense those pheromones of the queen, and instead of just walking all the way to the queen, they would stop at a certain distance, start fanning and scenting. Scenting is the jargon name for that behavior of producing the pheromones. And then another bee would come from behind, sense those pheromones that were amplified by another bee, and do the same thing. So they create this really interesting network that I'll show you some experiments of. So we wanted to understand how the bees do this. How do they make decisions about where to go, where to stop, and amplify the signals? And for that, we partnered with Greg Steffens, who is a professor at Orit and at the University of Amsterdam. Greg's lab helped us construct a high throughput classification code, basically using machine learning, where we were able to train it with lots of movies of bees that are scenting and bees that are not scenting. And basically the output would be, is this bee producing pheromones and scenting behaviorally? And if yes, what is the direction of the scenting? So now we can combine that method with our experiments. And here is an example of that kind of experiment and also analysis. So in our experiments, it's a semi-two-dimensional arena. We're looking at it from the top. It's an aerial view. The queen is placed here at one of the corners in a little cage. So the bees can still smell her, but the queen will stay more or less stationary in this area. And then the rest of the bees are positioned at the other corner of the experimental arena. And then every time you see one of these blue arrows, that's a bee that is scenting. You can also see the direction in which it's transmitting the information. Okay, so here is an example of one of these experiments. I'm going to play that movie. You can see that starting in the process, there's a lot of bees that are scenting. There's a lot of blue arrows. And if I scroll a little bit faster through the experiment, then towards the end of the experiment, after about half an hour, the bees find their way to the queen and they aggregate around the queen. Okay, so the first thing that we wanted to check is, are these scenting events really create some kind of directional signal that points bees towards the queen? So what we did to illustrate that is, and this is illustrated here in this cartoon, we basically took all of these scenting events and scenting directions in each one of the frames of the experiments. And if they are pointing, in some direction that is supposed to show us where the queen is globally, then if we integrate all of these scenting events, treat them as gradients on a higher dimensional surface, then higher values on the surface should correspond to where the queen was in the experiment. So here's just one example from one of these experiments. This is the reconstructed surface on the right side, and we see that higher values of the surface do correspond to where the queen is. The bees, by the way, don't always find the shortest path to the queen. I think in the experiments that I showed you before, they kind of go upwards and then right instead of just diagonal. But that's exactly why we did this exercise, to make sure that eventually the bees do point to the queen. Okay, so that's just proof of concept. And then we dived a little bit more into the experimental details to see if we can figure out what is triggering the scenting behavior. So from that, we have two hints from the experiments. We know that the number of scenting bees, which is what I'm showing here on the y-axis, is going to be proportional to the total number of bees that we have in the experiments. So we ran some experiments where we had different number of bees in the experimental arena, and we can see that the number of scenting bees is proportional to the number of bees that we have in the experiment. So there's some kind of probabilistic decision making about whether to scent or not. The other thing is a little bit more detailed in spatially. So we noticed that when we measure the distance between bees that are scenting and their neighboring bees that are scenting, we get a pretty consistent length, which is about 6 centimeters, besides one outlier we had in very small densities of bees. And that's interesting because if the bees are using pheromone communication, we know pheromones follow some kind of diffusion equation. And if they get triggered at a particular distance, usually a particular distance also corresponds to a particular concentration. So maybe there is a concentration threshold that triggers that behavior. So what we did next is we took all of this information we had from the experiments and use it to build an agent-based model where we can more explicitly change some of the behavioral parameters and understand the decision-making of the bees. Okay, so let me orient you here. So we have a two-dimensional simulation arena. The bees tend to move in a random direction unless they encounter a spot that has a concentration above a certain threshold. And then here comes the probabilistic approach. 50% of the time they would just walk up the pheromone gradient towards higher concentrations. And then the other 50%, they would generate their own pheromones. And they can do this either in an isotropic way or in a directed way. And we have that as a parameter in the simulation. It's kind of like a knob that we can change. All right, so we can put this into the simulation and run a parameter sweep where we change some of the behavioral parameters. For example, the concentration threshold and also the directional bias. And we see a couple of different phases of the aggregation around the queen. Let me just go through one by one. Excuse me. So the first phase is that when the concentration threshold is too high, there's no clustering. That's not surprising. When the concentration threshold is too low, the bees get stuck in local minima. Also not surprising for some of these aggregation problems. We also have at intermediate parameter regimes, diffusion limited aggregation. And then there is a sweet spot where we get these percolating clusters. Let me just play this movie. You can see that the bees do find a way all the way to the queen. So again, this is not super surprising for aggregation problems. But I think what's really interesting here is that there is this additional regime, additional phase where we get more efficient aggregation around the queen. And that only happens when we have directional bias. So it could explain why the bees use that directional bias and why it's more efficient. Okay, so the last thing that I want to say about, is there a question?
Orit, yeah, I do have a question. It appeared from the data that you showed before, eventually the bees terminate scenting. They eventually stop scenting. Is that correct?
That's correct. And that's actually not something that we have currently implemented in the model. So they just continue scenting forever. We know that's not realistic. And we don't really know what gets them to stop scenting. But if you have some ideas and a way to, it would be really interesting to include that also in the models.
Yeah, I'm just kind of curious if, for example, you introduce sort of a concentration, like a bee density or a pheromone concentration threshold on terminating scenting, whether it affects the qualitative phase space.
So the pheromone concentration, I'm not sure it would work because at the end of these simulations, the bees are actually aggregated around the queen and there's lots of pheromones here. So using that as a signal that might be problematic, but the density is a really good idea. So once the bees are surrounded by lots of other bees, maybe that would be a good time to stop, but it wouldn't deal very well with this kind of situation where you have small aggregations that are just not located around the queen.
Right.
Yeah. So it's an open question. If you have other ideas or if you want to try to implement something like this, I think it would be really cool.
Can I ask a question about this directional bias?
Yeah, please.
I probably just missed this, but how does I guess it work both in experiment and how does the experiment correspond to the simulation here? Like what's the
Oh, so yeah. Are you asking if we know what is the physiological value of that bias?
Oh yeah, a little bit. Or what it even corresponds to, like what the bee is doing that.
So what the bees are doing, they're fanning their wings. This is, I think in one of the movies that I've shown before, you can kind of see them flapping their wings. That's what's creating the directionality in the first place.
Because they're flapping them in a certain direction. Because they're flapping them forward or something, and they're
exactly
pointed forward.
they're pointed forwards, they're flapping the wings, and they're driving air backwards, basically.
Right. Okay.
Yeah, so there's a few measurements with individual bees showing that mechanics of that airflow, and this is how we know it. But there is almost no information about the actual diffusion of the pheromones and how long they reach, and that's why we left it as a behavioral parameter in the simulation.
Okay.
Yeah.
Cool. So it's like, I guess is it like that that it hits off the wing in a certain direction, and it's not clear how the pheromone, because I guess you can measure the direction, you can measure the polarity of the bee or something, right?
Yeah, and we know the body orientation of the bee. I guess what we don't know, I should actually go back here. We don't know anything about the initial concentration. We don't know anything about the diffusion coefficient. And so we don't really know what is the shape of that plume.
Okay. Cool. All right. Thank you.
Yeah. And it would be great to know, but it's very hard to visualize pheromones because they're so volatile and it's hard to tag them with anything.
I see. Interesting.
Yeah. All right. So the last thing that I wanted to mention about dense aggregations is that we just saw some examples with bees. And there's actually some really interesting, it's just a whole zoo of invertebrates that aggregate. And I want to highlight a few of them. And I think there's a really interesting opportunity to think about the transition between more spherical individuals and more elongated individuals that are a little bit more like, entangled polymers. So let me just show you some examples. We just talked about the bees. They're a little bit more spherical than other creatures, for example, gnat larvae, and also earthworms. So that's another project that we worked on recently in the lab in collaboration with Saad Bamla and Dan Goldman from Georgia Tech, who do really fantastic experiments with these earthworms. So let me just show you an example of that. So this is it's called a blob. That's the name they gave it. So it's a warm blob. What is happening here is that there's actually a temperature gradient, and the blob is moving towards the colder side of the experimental arena. They also do other things. They sense food sources, and they kind of go through these interesting phase separation transition, very similar to what we know happens in polymer materials. So just for fun, we decided to try to use a polymer physics model to try to mimic the behavior and also try to understand some trade-offs in the decision-making of the worms. So very briefly, we used a worm-like chain, and it's ironic that they call it worm-like chain. These are actually worms that has some bending stiffness, which we tune based on experiments Saad and Dan did with individual worms. And then we have some attraction between the worms and essentially also some tendency to move in a particular preferred direction. So we can get, replication of the behavior of moving towards colder temperatures. And again, kind of like we did before with the bees, we can run more simulations with different behavioral parameters. For example, the active force that the worms extract towards the preferred environment, or the attraction between individual worms, and we can see some interesting trade-offs and optimality in these behaviors. Okay, so just to summarize that part, and I'm running out of time, but hopefully that's okay to run off a little bit. So we've seen a couple of interesting examples of dense aggregations. Talked about how aggregations happens in the first place for bees, how they respond to environmental perturbations by changing the shape of the aggregation and also potentially moving collective locomotion towards preferred conditions. Which brings me to the second part of the talk that I prepared. And I want to make a conceptual switch to talk about more sparse swarms and how individual animals can potentially synchronize their internal states. So that's going to be focused on fireflies and synchronization models of fireflies. Okay, so a little bit of background. Fireflies are a very, very understudied organism in comparison to what we know about honeybees that are domesticated and very well studied, easy to work with. Fireflies, there's definitely less information out there available about the behavior. What we do know is that they have an organ at their abdomen. It's called a lantern, producing cold light, bioluminescence. And they use that to communicate with each other, mostly from what we know in the context of mating. So during the mating cycle of fireflies, they would aggregate in these very large swarms, and each species would flash at a particular frequency or particular pattern that corresponds to that particular species. So each species has its own flash pattern. And the males would flash, the females would respond, and that's basically how they facilitate communication during mating. So there is it is a complex signal. It's definitely, there's interesting information potentially in the onset of the turning the light on and off. For the purpose of that talk, I'm just gonna coarse grain it into a almost binary signal of on and off, thinking about it as potentially just visual signal of communication. And I'll show you in a couple of slides that we can actually trace fireflies and how they communicate in very, very large groups. So going back to some questions about communication strategies and how to best communicate in large groups, I think this is a particular problem where we can think about detectability, so high signal to noise ratio or compression of information, and it's sort of well defined because the signal is so simple. We can really just try to quantify it. And thinking about signal to noise ratio, fireflies definitely need to deal with that problem, and they seem to be addressing it with two different approaches. One approach, which is the movie that I'm playing here on the left side, is synchronization. So if all the communication channels are producing the same signal at the same time, then that's going to help with the signal to noise ratio. And there's actually a whole other class of fireflies, firefly species who don't synchronize their flashes. And as you can see in this movie here on the right side, even here in this movie, just by eye, it's very hard to know which flash came from which individual, right? It's hard to connect the dots, which is making it hard to know what was the flash pattern that this individual flashed. So we also do some work in the lab on these kind of patterns, but for today, I think I'm just going to focus on the synchronized species.
Orit, just a very naive question. Is the basic communication task here for a male firefly to say, "Here I am," and the female to like sort of help the female find the male, or is?
That's what we think, yeah. So with the swarms that we work with, the females are on the ground. They don't flash very often. It's actually very hard to find them. Mm-hmm. And we see a lot of males flashing around, kind of flying around a little bit above the ground, and they produce the majority of the signals that we see. And then if a female likes that signal, then she would respond with also a species-specific pattern.
Is it, I guess it's not clear to me the role of the female flash here. It could just be for the female to fly towards the male. So how do you view that?
So the females actually tend to stay, they don't move very much, even when they communicate. So what's happening is that the males would land close to the female
Oh, okay.
And then they continue the conversation, where both of them are stationary.
So in some way, selection is happening where the female is looking at a male's pattern and also deciding whether to flash back, perhaps.
That's what we know from the literature. Yes.
I see. Okay.
Yeah.
Okay.
Yeah, and actually they do this funny thing where they the females would actually direct their lantern towards the males that they like, so it's possible to know, also which one they're talking to.
And is there any understanding of what they're looking for? Is it like brightness or some kind of ordinal thing like that?
Yeah, there's not a lot of information. There's a few papers showing evidence that females of some firefly species prefer punctual males, in the sense that they produce a pattern that is similar to the stereotypic pattern of that particular species. For synchronous fireflies, there's also some elements of the group. So females would tend to respond when the males are synchronized and when there's more males. But it's not, I think there's still so many open questions about that process of decision making of the female that I would love to know.
Yeah, absolutely fascinating. Ben, why don't you go ahead?
This might be sort of a silly question, but in the case of the asynchronous blinking, is that actively maintained, or are they just randomly blinking?
So no, they actually use their species pattern. So usually it's some kind of burst.
Are they like intentionally avoiding one another though?
Yeah, we have no idea. And we actually do work with some non-synchronous species, and yeah, are they trying to just space it out to avoid each other? That's an interesting idea. We haven't tested that yet.
Sure.
But
it's kind of hard to measure.
Should. Yes. Yeah. Yes.
I had a question. So, like, why do they need to synchronize? Like, if it's just a mating signal, like, why shouldn't shouldn't the individuals be just doing it independently of each other?
Yeah, exactly. So we know they synchronize. We don't know why. There's lots of hypotheses as to why. One of them is that kind of what I mentioned about communication channels. If there's females who are far away from the swarm, if the males are synchronized, potentially that would increase their visibility just as a group to females who are further away. It's called the beacon theory. There's other theories, for example, actually related to the female's role. So some of the species that we work with, they flash in bursts. So when the males are synchronized, there's a very well-defined burst, and then there's a quiet part, and the females would usually flash back during the quiet part. So another hypothesis is that maybe by synchronizing, the males prevent or help the males prevent any kind of noise that would occur while the female is flashing back. So it's helping the female with the signal to noise ratio. And there's more ideas. So, but we really don't know.
Thank you.
All right. So we've seen that. Okay, so I'm going to focus on the synchronous fireflies. I'm sure you all are familiar with models of synchronization in non-living systems, like these pendulum clocks from the famous story, and also in lots of living systems. Even us humans, we synchronize our behavior. In some parts of the world, there's a tradition to synchronize claps at the end of a concert. Not in America, not in the US. I don't know why, but it's very interesting to experience in other parts of the world. Musicians also need to synchronize when they're playing musical instruments, and then fireflies are kind of one of these landmark examples that I still remember reading about in textbooks as an example of synchronization in nature. So this slide is really just to illustrate that there's lots of different models that can explain the emergence of synchronization based on pairwise interactions. Some of the more famous ones are the Kuromoto model that assumes that each individual is performing some periodic function, and then you can couple the phases of that periodic function between pairs of individuals. There's lots of different ways to implement that coupling. It can be a constant. I'm just showing here different variations of that. It can be a function of distance. It can be many different things. And the point that I wanted to make is that all these different kind of coupling would result in slightly different collective behaviors, but there's very little experimental data, empirical data to validate some of these models. So that that was the start, and that's the gap that we were hoping to fill. A couple of years ago, Raphael, who's a postdoc in my lab, and Julie, who was a student, we decided to just drive to the Smoky Mountains, where we know that there's large swarms of synchronous fireflies. These are their species name is Photinus carolinus, and they're one of these species that flash in bursts. So they flash on off a couple of times, and then quiet, and then they repeat that for a couple of hours every night. So here is an example of a recording that I hope is playing well. Hopefully you can see some flashes, and now it's dark, so now it's the quiet part. And here is another burst starting. And actually, I don't know if you can tell, but it's not really perfect synchronization. I think one of the things that Raphael told me in hindsight is that he was really worried the synchronization is just going to be perfect, and then there's nothing we can learn about it. So we're kind of relieved that it was a little bit more messy than what we imagined. But anyway, so this is one camera view. What we get here is basically a 2D projection of the entire volume that we're capturing. So we basically get temporal information. And we can see some interesting things from that temporal information. For example, when we plot the number of flashes as a function of the time, here just showing 40 seconds at a time, we see that when there is low density of or low numbers of flashes, there isn't any obvious pattern. But then at high values, high numbers of flashes, on average, or it potentially corresponds to high firefly density, we see these bursts emerge as a collective behavior. And we can actually characterize this a little bit more properly. For example, we can look at the variance of the values of these number of flashes over a window of time as a function of the average number of flashes. So low values on the x-axis here correspond to less flashes, and high values correspond to higher density of flashes. And we can see these two different regimes where at low densities we see something that is has a scaling law of a Poisson distribution. So it's in a sense a random process. And then at higher values we see that we're getting these clustering distribution that is characteristic of the burst structure that we see. Okay, so that's temporal information. There's more we can learn from it, but I want to show you that we can also actually capture spatial information, which is potentially important for some of these mathematical models that consider the fact that if fireflies are at a particular distance from each other, that distance might eventually also modulate their the strengths of the interaction, and then beyond a certain distance, maybe they cannot see each other. So it's actually important to try to characterize that. Yes, Ben.
Can they see the flashes of other fireflies while they are lit up?
Oh yeah, very good question. So we don't know. I would say that physiologically speaking, the lantern and their eyes are positioned as further away from as possible, on their bodies. But we don't know. All right. So to get spatial information, we need to employ more than one camera. We need the stereo vision, 2 cameras at minimum. So that's what we did. We brought a couple of cameras with us. We had the more traditional version of stereo vision, where we had 2 cameras kind of looking at the swarm from the outside. And we also have a version of this where we use GoPro cameras that have 360 field of view. And we position them kind of in the middle of the swarm. So we get, in a sense, a little bit the perspective of what a firefly in the swarm would also experience. And I would just say that these GoPros are super easy to operate. And because of that, we have a project where we are trying to actually crowdsource collecting information about fireflies, because really all you have to do is go with 2 cameras, press record, and then we have some automatic code to extract the information. Okay, so here's an example of that reconstruction, the information we get after we do the triangulation. So what you see here, that's one burst. Now it's going to get quiet again. Then we see another burst. And we can potentially record this over the duration of the night, over a few hours where the fireflies are flashing. So we get some interesting statistics about the spatiotemporal correlations of the fireflies. For example, here is one of these spatiotemporal correlations where this is a histogram of all the pairs of flashes. And the y-axis is how far away they were from each other in space, and the x-axis is how far away they were from each other in time. So actually these red blobs here that kind of increase in height, they're the signature of an individual firefly, because they're also flashing in bursts. So there's going to be some correlation just based on the fact that individuals produce more than one flash. And then we get this correlation between pairs of fireflies that are typically, at a distance of something like up to 10 meters from each other. And then that correlation decreases. So, maybe it tells us something about their perceptual abilities. Maybe it tells us something about the model of communication, synchronization. We're not quite sure yet. So now, just the next steps would be to try to use that information and plug it back into some of the mathematical models that we know about synchronization, and introduce that spatiotemporal correlations. That's what's happening on a fairly short range. I want to show you that there's some other kind of spatial correlations and propagation of information on larger length scales. In particular here, what I'm showing you is the colors correspond to the onset of the burst. So blue, the color blue corresponds to flashes that occur earlier in the burst, and red occur later in the burst. And what's happening here is that we have this kind of elongated field of view. This is all the flashes that we recorded in that field of view. And then here where my cursor is, we're just looking at it from the top. And what we're seeing is that the flashes tend, at least in this case, they tend to get away from the camera. And that's not always a direction that the signal is propagating. It actually changes quite often. But we can get some statistics about how fast that information is propagating. And it happens to be tenfold faster than the speed of the flight of individual fireflies. So the movement of fireflies, the fact that they're moving, cannot really explain in completion this this propagation, and there's probably some kind of delay in the response of individuals that might explain that propagation. Okay, so I'm over an hour. Just a question to the organizers: how fast should I run through the rest of my slides?
It's up to you. If how many more slides do you have?
Yeah, maybe 10 more slides. I can be strategic about what I show you, if that helps.
Yeah, absolutely. Why don't you keep going?
Okay, so everything I showed you about the fireflies so far, these are just fireflies in their natural environment. We can only record a small fraction of the swarm. The swarm is actually ginormous. It spans miles over miles, and we never see the end of it, honestly. So we don't really know how many fireflies we have in the field of view. We don't know exactly who they interact with. So for that, we also brought some more controlled environments, which are really just big black tents that we bring with us to the field. We catch fireflies and we put them in the tent. And we can see some interesting behaviors of the fireflies. In particular, if we look at the time between the onset of one burst and the consecutive burst, we see that the distribution of these times heavily depend on how many fireflies we have in the tent. So here at the bottom, that's the distribution of these times for relatively high number of fireflies, 20 fireflies. So we have a peak around 10 seconds. That's also what we see outside with a larger swarm. But as we reduce the number of fireflies to 5 fireflies, this distribution becomes wider, and it's becoming even more wide for 1 firefly, which tells us that maybe we need to adjust some of the constraints or some of the assumptions we make about the behavior of individuals. Maybe they're not best described by oscillators, maybe something a little bit more flexible in that sense. So this is where some collaboration with Shri Iver Biswas and her student Kunal came into place. And we actually used theory and computational modeling that is relying on models that were originally designed to actually simulate neurons and their synchronization activity. So these are integrated fire neuron models. So I think I probably shouldn't go too much into details, but I just say that this is one way to release the constraints about the periodicity of individuals. And by running these models, we can see the emergence of a collective time for that period. And if you have questions, I'm happy to go through this later in the question section. The last thing that I want to say is that we can also do some experiments with external lights, and we can influence the behavior of the fireflies using LEDs. So we basically bring in this little setup with an Arduino. We can program the LED to be at a particular frequency. We can record how the fireflies respond. We can essentially entrain them in some conditions, and we're not unable to entrain them in other conditions. As I'm sure you know, this is a nice way to try to learn something about the internal mechanisms. Is a fire an oscillator? And if yes, what is typical frequency, and so on. That's all I have for today. I think I'll just put in the slides to just thank my amazing team. This was definitely a collaborative effort of postdocs, graduate students, beekeepers, undergrad students, high school students, lots of amazing collaborators and funding foundations. And yeah, so it's been a pleasure working on it. I'm happy to take any questions, and I think I'll just stop here.
Thank you so much. Really amazing. I guess we could take some questions that are pertaining to the research content before we move on to the broader discussion. I can start with one. So I guess, is there any evidence that, so going back to the first part about the bee swarms, is there any evidence that the mechanical properties of the stable bee swarm is tuned to sort of the natural perturbations of the environment? So like, it would seem like on a tree branch, maybe the vertical motion is much more prevalent than the horizontal lateral. So is there evidence that, the stability of bee swarms is more attuned to this, or not?
Surprisingly not, or at least I would say we don't really know what are the typical perturbations that they would experience with on a tree branch, and this is potentially measurable by tracking where bees land. I would just say that they spread out, they respond like I showed you when we shake them actually right and left. They don't respond at all when we shake them up and down. And they actually break. So here's a movie showing that breakage. Same perturbation, exactly, just same perturbation, same swarm, but we're shaking them up and down. So we're not sure why, but that's just something that the bees do.
Yeah, it's very odd. I guess like one thing that I found a little bit striking about the videos that you showed before with comparing the natural to the experimental is that the natural seemed a bit stiffer. I'm not sure if I'm making that up, but yeah, it seems as though yeah, okay, so I see very little jiggling there. It looks quite rigid, right? So I don't know. It seems as if the swarm should be attuned to this kind of motion, but it's very odd that they seem to break up.
I agree.
Yeah. Okay. Avanish?
Yeah, on that note, do you think, I mean, they're able to sense the wind to some extent, right? Could there be a feedback process where they are able to use that information?
So if bees on the surface sense the wind direction somehow, and they tune the response, or at least integrate that information also with the mechanical forces that they feel, is that what you're suggesting?
Yeah, I guess the central question I was wondering is what is the timescale of their adaptation, and if they can, because it's a huge collective, right? So if they're able to like incorporate environmental noise into the adaptation of their structure and function, would they be able to? I guess that's the question.
So their response time is much slower in comparison to the time of the perturbation, at least that we applied in the lab. So we applied shaking of 1 hertz, 5 hertz, maybe a little bit slower. So at the order of a couple of perturbations a second, and it actually takes them about 15 minutes to 30 minutes to completely, to complete the morphology change. So potentially, they have also the privilege of sensing lots of environmental perturbations and respond to them in some kind of integrated way.
I have a question about the fireflies now. So you described this the crowdsourcing effort. It seems like that would be a really nice way at observing variations in behaviors that occur in different seasons or interannual changes. Are you thinking about these kinds of questions?
Yeah, so I haven't mentioned this here at all, but actually the tracking the same swarm over multiple years has some potential applications for conservation of fireflies, which have been neglected until very recently. So in the last couple of years, there's been a few groups who started working on that. And there's lots of firefly populations who are declining. One of the main stressors they have is actually light pollution, climate change. So we hope to also use that information for try to monitor fireflies in a high throughput way. And then, there's lots of interesting behavioral questions that we can ask. One question that I don't know if, it's maybe a stretch, but could it be that the same species of fireflies that we know on paper by the literature, they have the same, they should have the same flash pattern? Is there any environmental factors that could drive them to have slightly different patterns, dialects, if you will? That would be, this kind of crowdsourcing could be a really nice way to try to address it.
Yeah, absolutely.
Somewhat related question. Do you know much about the behavior of the individual fireflies? Like, can they migrate between different giant swarms, or do they move around a lot within a swarm?
No.
I mean,
we
don't
know.
How do you track a single bug?
Yes. Yeah. And actually, I should point out that we can see them we can track them as long as the time between flashes is short. It's below the certain distance that they would typically travel, giving their speed of flight. But for example, for the ones that flash in bursts, they have this quiet part, and we completely lose track of everybody during that part, which is a little annoying.
Okay, if there's no more research questions, I'm going to stop the stream now.